ICOE meeting roster, so please, everyone, whether or not you are actually members, please sign in, but indicate whether you are members.
We'd like you to be members, of course.
Good afternoon. Welcome to this seminar, which is jointly sponsored by ECE Interdisciplinary Distinguished Seminar Series and ICOE ENCS SP chapter. And we are very fortunate to have Professor John Mathews from University of Utah, who's currently a distinguished lecturer of ICOE's Neuroprocessing Society, to give us a very interesting talk here. Dr. Mathews' interests are in nonlinear and adaptive signal processing and applications in audio and computing systems, biomedical engineering, and structural health management. He chaired the department of ECE at Utah during 1999 to 2003. He's a fellow at ICOE, and he served as vice president on finance and then on conferences of the Signal Processing Society in the past years. He also served on editorial boards on various journals and magazines in the Signal Processing Society. So let's welcome him.
Thank you so much. I was glad to hear that you're saying that this is a very interesting talk. Now I'm happy. I'm satisfied with my talk. I can go home, right? So what I want to talk about is an area where I think we signal processors can do a lot of transformative contributions, restoration of motor skills in patients with disorders of central nervous system. Essentially, what we are talking about is if you have a spinal cord injury and if you are paralyzed, can you give the movements back to the paralyzed limbs? Those are the kind of questions that we have been asking, and we are not the only people who are asking those questions. There's a lot of activities going on in this area. So you will hear a little bit about some of the things that are being done in Utah in this area. And so let's just go forward. Okay. I guess this video is not very good, but I think the other screens probably are okay.
This needs a lot of signal processing.
Yes.
To de-blur that.
Absolutely, absolutely. But I am assuming that you are all watching the other screens. So what I want to do is to start with a video, a short clip, about 90 seconds or so, that came from a 60-minute program, actually, from CBS. That video was about 13 or so minutes long. You can do a search for robotic limb controlled by the mind on CBS news sites, and you will actually find the video. I just extracted a few seconds that are relevant to us from that. So let's just...
Who wouldn't want to do this? When they told me I heard about the study, I said, oh, absolutely, I couldn't not do this.
Last February, Jan Sherman put herself on the line.
What I'm going to do right now is I'm just making some marks here in your hair.
The procedure was done by University of Pittsburgh neurosurgeon Elizabeth Tyler Kavara, who showed us that the area that controls hand and arm movement is accessible right on the surface of the brain. During the 6-hour surgery, 2 sensor arrays, each the size of a pea, were placed on the surface of Jan's brain.
Woohoo!
Then they were wired to 2 computer connections called pedestals, the gateways to Jan's thoughts. 5 months after the surgery, we came back to see whether she would be able to control the robotic arm with nothing but her thoughts. They plugged her brain into the computer, and this is what we saw.
I can move it up and straight down, and left and right and diagonally. I can close it and open it and I can go forward and back.
That is just the most astounding thing I've ever seen. Can we shake hands?
Sure.
No, really.
Yeah.
Like, come right over here?
Yes, you come over
Okay.
there. Let me grasp your hand there. There we go.
Oh, my goodness. Wow.
And I can do a fist bump if you'd like.
That's amazing.
Okay, so this lady, Jan Sherman, she had a degenerative neurological disease, so she is paralyzed from neck down. A couple of years ago, she allowed herself to be implanted with a couple of electrode arrays in the brain, hoping that she could use the signals that comes out of these electrode arrays to control robots to do things that might actually help her. Obviously, this is experimental stage. She is doing this for the future generation rather than for her own. But what this short video showed is probably the state of the art in terms of what brain-computer interfaces can do for in the context of assistive devices. A robot assisting a paralyzed person to do the things she or he is not able to do at this time. Okay? What we are trying to do is complementary to this thing in the sense that a part of the things that we are trying to do is to actually make the limbs themselves move. Okay? It's a little different. So let's move on. By way of introduction, in the United States, there are about 12,000 new cases of spinal cord injuries each year. About 50% of them come from automobile accidents and the rest from other causes. Okay. There are other causes of paralysis, strokes, brain injury, multiple sclerosis, etc. Okay. It's estimated that in the United States there are about 6 million or so people who have some form of paralysis, not complete paralysis, but some form of paralysis. So what that means is that chances are that all of us know somebody who is suffering from this condition. Okay? And it's those kind of people that we are trying to help. Now, before I start explaining physiology to you, I must tell you that my understanding of physiology is not all that great. Usually I go through these things, I know just enough to explain these things, and then I find that there are people in the audience who know much more about physiology than I do, and when there are questions about physiology, usually you get better answers from the audience, and I go away learning more from the process. Okay? So let's just talk about how we go about trying to treat this disease. Spinal cord injury is a disease, so I'm going to just call it a disease and go on. Okay? So in order to do that, we need to understand a little bit about human physiology, the physiology of nervous system. And me being an electrical engineer likes to actually work in block diagrams. So we will do block diagram level representation here. Okay. So the nervous system basically is consists of two main parts. One we call the central nervous system, the brain and the spinal cord, and then the peripheral nervous system. Okay? The peripheral nervous system basically contains a whole bunch of nerve fibers that innervates all of the body. Okay? It contains both the motor nerves that would excite, induce movements, as well as sensory nerves that will actually sense the state of the body, in each location. Now, these motor fibers would communicate with the central nervous system using discrete electrical signals. We call them action potentials. All right? And essentially what happens is once the brain, the central nervous system, gets information about the state of the body, it does some computations, it will actually make some decisions, it will send orders to perhaps the motor nerves to do something. Okay? So essentially it's a communication system. All right? Now, when you have a spinal cord injury, what happens is this communication system gets broken. Okay. So how do you treat the problem? You have to somehow restore the communication system. If you have to do it artificially, we will do that. Okay. So that's the process that we are actually trying to accomplish. Before I tell you what device we might use to accomplish this replacement of the communication system with something that is artificial, let me talk a little bit more about how the nerves are organized in the human body. Okay? So these nerve fibers are actually usually formed into bundles. These bundles we call fascicles. Okay? So, and there are multiple fascicles you will find in a nerve. All right? And this fascicle contains nerve fibers, both sensory and motor type. And they actually affect all parts of the body. All right? And the notion is, can we access these nerve fibers in a selective manner such that you can sense what is happening in areas corresponding to where the nerve affects you? All right? Or can you give instructions to the nerve to do something? Stimulate the nerve so that it will actually move the muscle or create a force, something like that. Okay, that's what we're trying to do. And what we do is we do that with what we call the Utah Slanted Electrode Array. The Utah Slanted Electrode Array, the Utah Slanted Electrode Array, this is a 4 millimeter by 4 millimeter array of 100 electrodes. All right. And it has one other interesting issue. The electrodes here are much shorter than the electrodes here.
Is it made in Utah?
It is made in Utah. It was invented in Utah. It is made in Utah. And almost everybody uses that too. Okay. Well, almost everybody. There are other kinds of electrodes that are there. There is a Michigan electrode that people use. We like this one much better. Okay? So, there is a company actually that builds these things in Utah called BlackRock Systems right now. And they sell it to almost all the educational institutions that are actually doing this work. So anyway, so what we're trying to do is this is the device that's going to bridge the gap that is created by the spinal cord injury. Okay? So let's talk about inserting or implanting an array like this into the nerve. Okay? There are two things we need to understand. One is the spacing between the electrodes are smaller than, in general, the size of the fascicle. So you can actually go inside the fascicles. You can be more selective than actually going to just one bundle. Okay? The second thing is because the length here is smaller than the length here, and by the way, only the tip, it's gold tipped, and then the rest of it is insulated. Okay, it's the tip that matters. Okay, so the shorter, if you're actually implanting out here, the shorter electrodes will actually penetrate till here. The longer ones will actually penetrate till here. So you have access to nerve fibers in the whole, all of the nerve. All right. So if you hear the term intrafascicular stimulation, basically what you're trying to do is you are going to go inside these fascicles and stimulate selected groups of nerve fibers. That's really what you're trying to do. Okay. So if you hear that term, now you know what that means. Okay. Now, these electrode arrays are implanted using a high velocity insertion method. There you go. You can see. So basically, just push it in, and you're done. Okay. So let me talk a little bit about selective recording via the Utah Slanted Electrode Arrays. By the way, you can see this name here. That's actually my daughter who happens to work in this area too. Okay, so I steal slides from any place I can get, including from my daughter. So this is actually trying to show the selective nature of the recording of information from the nerves that is possible with these electrodes. So what is happening is one of these electrodes, in this particular case actually, we have a high density electrode which is instead of 4 millimeter by 4 millimeter, it was 2.5 millimeter by 2.5 millimeter. Okay, that was implanted into one of the sciatic nerves of a rat. Okay, left sciatic nerve, I think. And what you're seeing here and here are the action potentials overlaid. There are about 1,000 of them in there. Okay. Essentially, it shows that these action potentials have a shape that you probably can depend on for some of the analysis that you're doing later on. That's one thing. But more importantly, what you want to actually see is, I wanted to look at this part. Okay. So this is an action potential coming from electrode number 21 in the array. This is action potential that's coming from electrode number 14 in the array. This thing shows you the time when electrode 21 was excited, and this shows you the time when electrode 14 was excited. And this shows you the activity that was happening at the time. Okay? So electrode 21 actually got excited when the limb was pushed up. Electrode 14 got excited when it was actually done the other way. Okay, plantar flexion, I believe that's called. Okay, so essentially what it's saying is that you can actually, by looking at electrode 21 and electrode 14, you may be able to say what was happening. The selectivity is a lot more, much more finer than this thing. I don't have the time, but I can actually show you examples where you would touch one location and one electrode will get excited. You touch a little bit later, after that you will get something else to excited. Okay, things like this. So it's very, very selective. That's good. So that's recording, sensing signals using this electrode arrays. Okay. Now I want to actually talk about exciting, stimulating nerve fibers. All right. So basically you're talking about activating motor units. So what you do is you apply electrical stimulations, okay, and what we do is we do constant voltage pulse. So basically we are doing a pulse width modulated signal, applying a pulse width modulated signal, okay, and we use constant voltage signals. There are people who use constant current signals. I'm not going to talk about what is better or the other at this particular time. This is what we started with, we never changed, okay. And so what happens is, when you apply a pulse like this to one of the electrodes, that electrode excites a small group of nerve fibers, all right? And then that results in some evoked muscle force. Typically the muscle force is in the form of a twitch, we call it twitch, okay? That's the form you will get. If you apply something that's a little wider, the force becomes a little large. All right. Now, if you apply to another electrode, another pulse a little later, so you get the original force, then this would evoke some other force. The shape can be a little different, okay, depending on which muscle group it excited. Okay. So the overall force might actually add up like that. Now, the overall force might add up in some nonlinear manner. We don't know for sure. I suspect it is. There is nonlinearity in it, but almost everything that we have done has assumed linear summative models. Okay, but none of those things are particularly important for this conversation at this time. Okay, so then instead of that pulse, if you have a pulse a little later, what would happen is the force evoked will come a little later. That means that the overall shape of the force over time will be a little different. So our task, let me just see. Our task here is to apply specific electrical pulses to different electrodes so that it can evoke specific motion, specific types of motion. Okay? And then our job is to actually find when do you apply the pulse to which electrodes, okay, and how much pulse width you need to have in order to get a specific force pattern output. Okay? So that's the problem. And that's just showing that overall muscle force can be controlled like that. So you know what you have to do, right? If you want to make a paralyzed limb move, you need to identify the nerves that control that muscles that control the joint or control the limb, okay? And you have to apply electrical pulses in some appropriate manner, okay? So let's see if it will work. This was done several years ago, so I just want to show you a video of, in this particular case, the cat's limb was, I think there were 3 different muscle groups that we needed to, yeah.
So the muscle force here, is it a signal you're measuring, or how?
We had a load cell that was measuring the force in that particular case. In that particular example that we're showing it was the force itself that we were measuring.
Force applied by a particular muscle.
Yeah, that's what we were measuring. And in that case, we could. Okay. Now, in many applications, what we are interested in is, let's say I have a joint to move, and I want to know what the angle of movement is. And maybe that's what we'll actually measure. But we have to have a model that tells you how that muscle force will change this angle. All right?
And how is the variation among, let's say, rats, like among different rats, how the relation between the input and the output?
Again, the way I look at this problem is it can be very, very different. Okay. The reason why it can be very, very different is when you do the implant, there is no guarantee that you will do the implantation in exactly the same way. Even if everything is precisely the same, there is no guarantee, right? But if you're thinking about applying this to a patient who is paralyzed, okay, you are going to do things that works for that patient. So you have to learn about what works, what electrodes you need to excite, what kind of signals you need to actually apply for each person separately. Okay, you're going to tune it to each patient. Make sense? Okay. So, yeah, I think there were 3 different electrodes implanted, and because of the number of joints involved in this, we are trying to actually just make the can stand up. Okay. Okay, nothing great, but hey, this was supposedly a paralyzed cat. Paralysis was induced actually by anesthesia here, so the cat was really not paralyzed. So I don't want you to think that we actually did some really bad things. Okay, but perhaps the movement was a little jerky. Okay, but we were able to induce movement in the muscles that was controlled by a machine outside. Okay, so it can be done. Now the job is, can you do more refined processes? Can you do these things better? So let me talk about one other thing. This is a video that's about 1.5 minutes long, so I'm going to start the video pretty soon. I want you to keep your eye on the 2 cats. Okay, it's actually 2 cats. It may have been, it's 2 different cats, I think. No, it's the same cat done at 2 different times. So we are trying to induce movements in 2 different ways. In this case, we use something called synchronous stimulation. I will explain that in a second. In this case, we use something called asynchronous stimulation. Our claim is you want to do asynchronous stimulation. Okay, and essentially you are going to do a series of stand up and down, sit, stand up and sit motions. Okay, you will find this one tiring out very fast and after some time not being able to do anything, and this one going on for a lot longer time. Okay, so I'm going to start the video and then I'm going to talk through it. Okay, not necessarily looking at. Okay, so the idea is you are creating motion using pulses, right? So if you want to get smooth motion, you should apply these pulses at fairly high frequencies. Okay? Otherwise, you can expect that there will be ripples out there. Okay? So what happens is that if you apply these pulses at high frequencies, muscles get fatigued very fast. Okay. So what do? What we can do is this. You have two options. One is you can apply the pulses to different electrodes at the same time. It is like applying electrical stimulation on a cuff electrode. If you have an electrode that does just cover the nerve somehow and apply a big voltage, it will affect every nerve inside. Okay? And it will do something in some overall manner, I don't know how, some integrated manner. Okay? Or you can apply a pulse to one electrode. After some time, you can apply a pulse to a second electrode, and so on and so forth. Okay? So if you go that way, we call it asynchronous stimulation. If you do the first thing, you call it synchronous stimulation. All right? Now, so what happens is with asynchronous stimulation, since you are applying pulses in an interleaved manner, it appears like you have a higher frequency than you are applying to each electrode. Okay? And because of that, the overall fatigue induced is going to be a little smaller. All right? And that's really what you saw out there. You saw this guy dying out much faster than this one, right? In fact, it was going on. You could see it actually weakening towards the end, but it was still going on at that time, right? So the claim is you want to do interleaved or asynchronous stimulation of these nerves. Okay? Yeah.
So are the people studying how the brain does the stimulation? Does it do it synchronously or asynchronously to our muscles?
I have no idea. I told you I don't know physiology. Okay? I'm not sure. I suspect that there would be something like this happening, but I don't know enough to say. Okay? So let me move on. So far, everything that I did was to just motivate the problem and to give you an introduction to how we might actually go about doing it. Okay? Now I want to ask two questions. You need to tell me when I should stop. Okay? So you have what? Another 20 minutes or so?
Two o'clock.
Okay. Yeah. Okay. Should be enough. So, yeah, I want to answer two questions. One is, how do you design stimulus signals to achieve desired movement? The second thing is, how do you know what the desired movement is? In the sense, how do you know the intent of the person? Okay. So let's just talk about stimulus design. So I have very few equations in this talk, okay, in the sense, Hamid, I know you don't use equations at all. So, so I'm trying to actually follow that paradigm here. So here is how we would actually go about the process of designing the stimulus in some iterative manner. You know, signal processors, we like to do iterative things. So that's what we're trying to do here. Okay. Now, so for the time being, basically what I'm trying to do is this. I'm assuming that I have selected 6 electrodes out of this 100 I have, okay, in some manner. And I'm assuming that I have somehow figured out what the first pulse ought to be and when I ought to actually give it for each of these electrodes. Okay. And so essentially the 6 different colors you are seeing are the responses from 6 responses to the 6 different electrodes. Okay. And the overall response is here. Okay. Now that you have gotten that, you can measure that response. You are ready to actually apply the next pulse to the first electrode. The thing you have to remember is this. The first electrode was most impactful somewhere out here. So if you try to actually make decisions about what pulse width you must give and at what time you must give the next pulse to that electrode, you must look somewhere here rather than here. You understand what I'm saying, right? Okay. That's very, very important. So you have to look a little behind because we are the effects are a little bit happened a little while ago. Okay. So that's probably where we should be looking at. Okay. And then based on that, you decide on what pulse width to give and keep doing that, and somehow we would get here. Okay. This was supposed to be the desired response that we were after. By the way, is a step response a right response to even work with? I know electrical engineers love step responses. You know, is a step a good thing to actually even try? What do you think?
No, probably not.
Probably not. Step. All right. So it's very relevant actually in this particular work. Okay. Because you're holding some force, right? You will see us doing sinusoidal signals quite often. Why? Because gait, when we walk, the motion is periodic. Okay? So some of the things that we love to work with are actually quite appropriate in this scenario too. So I looked at this problem and I said, and if you know me, you know that I'm an adaptive filtering guy. Okay? And I looked at the problem and said, this is an adaptive filtering problem. And I took one of my students and I said to him, just go design an adaptive filter for this problem. What do you need for an adaptive filter? Well, you need a system model. Here is the system model. The force evoked, I just said, is a sum of a linear combination of the responses due to the stimulus. Okay, from different electrodes. Okay. So I have a model out there. I need a cost function. What should my cost function be? Well, I'm going to look at changing the pulse width at this time based on what happened to the force measured at this time. Okay? So I have the measured force or evoked force, what I really want in that time period, whatever time period that is. Okay? I just did the squared error. Why not? It's better than nothing, right? So we do that. And then an LMS type adaptive filter, what do? Gradient adaptation, right? I said, update the amplitude change as the previous amplitude minus a constant times the gradient of the cost function with respect to the amplitude. Time offset, do the same thing. Previous time offset minus a constant times the gradient of the cost function with respect to that time offset. So, you know, you can do the math, you can do the implementation, it's straightforward. Are we done? Any problems with anything I said? It all looks perfectly fine, right? Except for one thing. I wanted to find the pulse width. I got the stimulus amplitude. Right? So what do? We create something called the recruitment curve. What it does is we actually characterize the electrodes and the interaction of the electrodes with the nerves before we do all these experiments after implantation. Okay? So we create a function a function basically that looks at the stimulus duration and the maximum peak amplitude of the evoked forces. Okay? So if you then say that this must be my amplitude, I will just look in that curve and say this must be my pulse width. Okay? So that's fairly straightforward. And this characterization has to be done every time you do an implantation, and sometimes you may have to recalibrate over time too. Okay?
So this is a characterization of the probes themselves.
Yeah.
Dr. Mathews?
Yeah.
Just one question. So what were the parameters L and K in the previous slide? The capital L and capital K?
Let's see. L here was the number of electrodes, I believe. And K was, I think, which pulse you're looking at, the response to each pulse, right? You're applying multiple pulses, right?
To each electrode.
Yeah. So you have, for example, aij, ith electrode, jth pulse. So let me just show you some experimental results. This was done on a cat. Left sciatic nerves because we are trying to move muscles in the leg. Okay. So we measured force evoked actually using a load cell, 6 degrees of freedom, load cell, and so on and so forth. In this particular case, the first set of experiments we were doing, we were actually constraining the muscles in such a way that it doesn't change its length. We were just trying to make things a little simpler. Okay, we call it isometric muscles. Okay, and this was just the machine that actually did the excited stimulation. All right, so we are ready to do the experiment. We have implanted the electrodes into the nerve. Okay. Before we do any experiments, or before we really do these experiments, we have to do 3 things. Number 1, we have to figure out which electrodes to use. Number 2, for each of the electrodes you select, you have to actually get the average twitch shape. Because if you actually looked at our cost function, I had a twitch shape out there. Okay? You can measure all those things. Okay? And which electrodes you pick, basically you actually stimulate each electrode, look at what kind of response it gets. We want to get different responses from different electrodes. We want to get fast response. And there are criterion that we had, and we don't want too much overlap. Okay? And based on all this criterion, we would pick a few. And almost every experiment I will show you, we used 6 electrodes. And that's not necessarily a number that you should use, but that's what we have done. Okay? And then for each electrode, we also need to find this curve that actually tells you how the stimulus width and the amplitude of the force evoked are related to each other. Okay. Once you have that, you can apply the math and implement the system. So, experimental results, I want to actually tell you one other thing. For this experiment, and for this experiment only, this was done 4 years ago, something like that. At that time, we did not have the ability to implement a real-time adaptive system. Okay. So this experiment, what we did is we simulated the situation. We got a sequence of pulses and sequence of times at which the pulses should be applied. And then we applied that pulse sequence in the real-time experiment. Okay. This is the only case where everything I'm showing is not implemented in real time. Everything is done in real time. The rest of the experiment, everything is done perfectly online, on time, in real time. Okay. So in this particular case, what we're trying to do was here is a step input or desired force response is the step. It's basically showing simulation versus the actual evoked measured signal is what evoked signal is what is happening. Okay, it works okay. The ripples are not anything is a little larger than I would like to have, and it actually turns out that we were doing 6 pulses per second to each electrode, effectively giving us about 36 hertz stimulation. Later on, every experiment we did at 60 hertz effective, and you will see a lot less ripple than this one. Sinusoid, there is of course going to be a certain amount of delay, and then some complex signal. Everything seems to work reasonably well. Okay, so let me move on. Then, after doing that part, we actually stepped back a little bit and actually did some work. And we realized that the adaptation of the time of asynchronous stimulation is probably not all that important. We still need asynchronous. We still need to find the right time to stimulate each electrode, but we can have the separation between each stimulation event to be the same. Okay, that's what we found out. Okay, so we stopped to adapting the time of the pulses, timing of the pulses. We simply adapted the amplitude of or the pulse width. Okay, so the first thing we tried was to simply try a proportional control for the stimulus width, and there you go. You see sine waves, you see much less ripple. Here is a more complex signal. You see fairly good tracking of what we want to do. Okay, nice. And fatigue resistance. So this was basically, this is what would happen. So this is about 5-6 minutes long. Okay, so we were actually asking the cat to hold a particular force for a long time. Okay. What would happen is, it's not that the muscles are not necessarily getting fatigued. It will get fatigued eventually. Okay, but since we have a feedback mechanism, we can actually sense the fatigue and apply more stimulus when the muscles get fatigued. And that's what we did here. Okay, and this is all done automatically, right? And basically, we said we are not going to increase the stimulus beyond this. 1 millisecond is what the maximum was. And so when we reach that point, you will start seeing the effect of fatigue. Yeah.
Comment at this point. It seems to me that it's looking at this from the physiological standpoint, fatigue is eventually going to set into the depletion of, say, ATP or glucose in the muscle. And what you're looking at here is what is the most efficient use of those chemicals by finding the right pattern. It's looking at it the opposite way from the way you are in terms of it.
I think you're right, yeah. Again, I don't want to argue too much at this particular point, but I think you're absolutely right.
But a muscle will eventually become fatigued.
Yeah, absolutely. And that's exactly what I said too. Muscle will get fatigued, okay, but this will allow you to compensate for the fatigue for a while. Again, not forever.
It seems like the dual actually sensing should be in place, right?
That could be done too, yeah, absolutely. Yeah. Okay, great. So now the question is, can we do this to a joint control? Now we have multiple muscles. Okay. It turned out that proportional control was not enough, so we went to proportional velocity control. We went to proportional integral velocity control. It turned out that that's the one that worked the best for us. So let me just show, this is an experimental setup where we are actually measuring the ankle movement and making sure that everything can be applied in a particular way. Essentially, you have the foot, you're trying to actually control that joint. There is a plate that actually would apply pressure, move the foot to leg to some angle, and then you tell the cat to move the plate back. Okay, that's what we are trying to do. So here is a video of what we could accomplish. And what we're trying to do is exactly that. You will see the plate moving the foot, then the cat moving the plate back, holding it for a while, and then going back. Okay, now you will see it going, pushing back. Okay. Overall, you could see a fairly good, smooth movement, but you could also see, actually, if you notice something here, you could see, shivering and things like that. The stimulation was actually also affecting some of the other muscles and so on and so forth. But overall, not too bad. Okay. This is just showing what happened with various types of things. A fairly high frequency signal. It turned out that we couldn't go all the way up, and we believed that it was mostly because we hadn't tuned these parameters that carefully this time. So the next question is this controller robust to variations in the operating environment? What happens if you change the step size in a step situation? Turns out mostly okay. What if the load is changed? Okay. Again, except for the very high loads, it worked out fine. What if there's a disturbance in the load in the middle of the process? Turns out, here we changed the load, came back, life looks good. Okay. What about stability over time? So in this particular case, we had done the implantation, did the experiment right after implantation, then waited 19 days, did the same thing again. Okay. Using the same set of parameters, not tuning, same set of parameters, same set of electrodes. Okay. Still, we got almost the same results. Okay. That's very, very comforting. Okay. Now, the response, these are the response. Now the question is this response time, the behavior adequate compared with what we expect from people? Okay. So what we did was we applied the same kind of load and all those things, the desired force kind of things, and asked people to actually push against the plate. Okay. So 4 people, and this was the machine, and these were the people. Machine is a little slower, but hey, this is the first time we are doing this kind of thing, so I think it's okay. Step down, the machine actually has a little bit more step-type responses, 2-step response. We know the reasons, but I am not going to explain it because it's actually based on some of the things that we did in the algorithm that can be actually changed. Ramp, again, machine's a little slower, but I think the key is it's not a lot worse than what we would expect people to do. Okay. So I have maybe another 5 minutes or so I'm going to actually quickly go and talk a little bit about decoding signals from the brain. So, first question, where to sense? Well, you know the brain is divided into different parts control different activities, different parts of the body. Somewhere out here, I think, is the motor control area. Okay, I think, I have no idea, but I think. Okay, I know that even in the motor control area, different locations actually control different parts of different parts of the body. Okay, so you have to sense from the right place. That's the only point I'm trying to make. What would you do? One of the most common devices people have used is EEG, electroencephalography. The good thing is it's non-invasive. The bad thing is you probably will not get very good signals. Okay, so we don't use that. There is little selectivity because there is too much attenuation, too much integration, everything going on. The other extreme is to actually have a brain surgery and put a penetrating electrode into the brain. All right. And I believe that was the kind of electrodes that was actually done for Jan Sherman, the first person that we saw. I believe so. Now, limited spatial coverage, but that's not a problem really because we have specific needs and we are going to do something there. Okay, but high specificity. So this might be good, but you need complicated surgery. So what we have done is we have actually gone to something in between, something called electrocorticography. Okay, ECOG signals. Basically, again, we have an array that you implant in the brain, but just below the cranium. Okay, so the surgery is not as complicated. Okay? So the hope is that there is enough information in the local field potentials recorded from through those electrodes. Okay. So again, because of lack of time, and by the way, the talk is designed like that because I don't give too much details in here. Okay. So essentially what we're trying to do is this is a patient who actually came to the hospital with epilepsy, okay, and needed some rewiring of the brain, so he needed a brain surgery, okay, and we asked them if we could actually implant this ECOG array also during the surgery, okay, and he would actually subject himself to some experimental process. During 1 week, he was there at the hospital. Okay. And after that 1 week, both the electrode arrays got removed, and we're done. Okay. We got lots of data. So we can work on that for years and years, and that's what the situation is. Okay. So in this particular case, so we have 32 channels from the brain that we can actually collect, and we were asking the patient to move the hand in. Oh, I thought I had put off the audio. So yeah. Anyway, so he is going to move the hand in different directions. Okay, and we are recording the movement of the hands as well as the signals from the brain. Okay, I'm going to move on. Yeah, you see what's going on, right? And now the question is, from the brain signals, can we track the hand? Almost everybody who works in this area uses Kalman filters. Most people use linear Kalman filters. Okay, we are going to use linear and nonlinear Kalman filters. Okay, we're going to do some nonlinear models in there too. Okay, so the key thing here is, when you talk about Kalman filters, you don't in real life, in real testing, you don't have hand signals. Right now we have hand signals, so we are trying to train the system, right? So there are several parameters in the Kalman filter that requires the hand signals. Okay? So what we're going to do is we are going to train for those parameters. We're going to learn what those parameters ought to be from the training data we have collected. Okay? And then we are going to test it by freezing those parameters and updating only the rest of the Kalman filter equations, okay, and on the rest of the data. So two things here. One is there is a delay between the brain signals and the hand signals. Brain should be ahead of you, ahead of the hand. Okay, and this is what this picture is showing. This is actually time delay number of samples. This one is for a linear. No, this one is for a linear model. This one is for a nonlinear model. It turns out that the nonlinear model is much better than the linear model. You would expect that, because there is no reason to believe that the brain is anything linear. Okay. This basically is actually showing there is how adding memory would help in the cause. Okay. 0 memory to a memory of 5 samples. Okay. For linear, and I'm sorry. Hold on a second here. No, I'm sorry. Yeah. This one is for 0 memory. This one is actually for a memory of 5 samples. And this one is actually using a polynomial, Volterra type model, polynomial nonlinearity, order of nonlinearity going from 1 for linear to 5 for 5th order nonlinearity. Okay. Again, you can see how well things work. So this is using 32 channels, linear models. Okay, the red is the actual hand movement. The blue is trying to actually track the hand movement. Not that great. Okay. Of course, I show linear models first because I have better things coming later. All right. So let me move to the nonlinear model. I think you will agree with me that this is much better than the other one. Okay, so you can actually see this tracking, and this is actually outside the training sequence. The hand signals were not included in the updating equations at all. Okay, it was trained on part of the data, but then it was frozen and then like that. Okay, so overall, not too bad, I would think. Then we said, do we need these 32 channels? Okay, so what we did is we started looking at the mutual information between the brain signals and the hand movement. Okay, and we picked the channels corresponding to the highest mutual information. Okay, if you just do one channel at a time, that's not very good, because you won't know anything about redundancy in the channels. Okay, so but if you want to do 15 channels, doing mutual information for 15 channels and to the hand signal is going to be very difficult too. Okay? So what we did is we actually did the mutual information between 3 channels jointly and the hand signals. Okay? And then computed all the mutual information in the signals, okay? And picked the channels that gave me the triplets that were the highest. Okay? So the highest triplets, I took all 3 of them. Okay? Then I went to the next triplet and picked, if one is different, I just picked that one, and so on and so forth. Okay? And this one used 10 channels. It's better than the linear case, worse than the using all 32 channels, but still not overly bad. Okay. So I'm going to move on. So that kind of concludes my talk. Basically, there are several challenges ahead. This is just getting started. Okay. Control for coordinated joint and limb movements. That's something that we still need to work on. Okay. Dealing with biarticular and triarticular muscles. We've just worked with one muscle at a time, basically. Okay. So many times, the muscle that control this joint also control this joint. Okay. How do we handle that? Okay. Another thing, there may be 2 muscles that are control one joint. When one muscle expands, the other muscle would contract, and they control things jointly. How do we deal with that? Things like that are ripe for signal processing processors to actually work on. Okay, these nerve fibers and electrodes change in time, change their interactions, change with time, okay, over time. Do we need to adapt to that? The brain actually learns about the situation. Do we need to adapt, or maybe that's fine? Brain will just take care of itself. Okay, things like that. There is much ongoing work still going on in this area. Beyond that, wireless electrode arrays, better model for muscle response, alternate stimulation techniques, optical, magnetic, neuroprosthesis for other functions. All these things are going on. Okay. Finally, let me just quickly say that this work is by no means something that I did myself. Lots of collaborators, including those 4 used to be faculty members in our university. Dick Norman is the one who invented the Utah electrode array. Bradley Greger was in our college, but now in Arizona. Greg Clark is in bioengineering. Dick was in bioengineering. Sandy Meek is mechanical. These 3 were our students. Mitch Frankel did almost all the stimulation work. Spencer Kellis did a lot of the Kalman filtering work. Almost all the nonlinear stuff I showed was done by Ehike Dantas. And that does my talk.
I think I have time for questions.
Yeah, so you said you were using square pulses for exciting the muscles, right? So
Pulse width modulator.
Pulse width, yeah. So how much does the exact shape of the signal matter in how the muscles respond?
The shape of the signal in the sense in the pulse itself?
Yeah, the pulse itself. Yeah. I mean, if you use like a Gaussian pulse or some other pulse.
To be honest with you, I don't know the answer. Okay, because this is what is traditionally done.
So because when you said you showed the signals you measured from the nerves, on the slide you stole from your daughter, the shape of the signal looked somewhat like a half of.
That was the response of the muscle. Okay.
So that's not the nerve signals.
Those are the nerve signals. I'm sorry, it's not the pulse that you are using to stimulate.
Right.
You are the action potentials had that shape. Okay. So that was recording. Okay. So recording something that happened in the nerves. Okay.
And that's different from.
That is different from stimulation. In one case,
it's on the same nerve, though, right?
Yeah, but in one case you are applying
Right.
stimulation that evokes muscle force.
Sure.
Okay. In the other case, you are looking at what the nerve generated to send to the brain, for example.
Oh, I see. Okay. So how do the normal the excitation signals look on the nerves which are being excited by the brain? How do they look like?
How do the normal excitation signal look?
The signals that the brain sends to the nerves, how do they look like?
I'm going to assume that they are more like the action potentials.
They're actually the action potentials themselves, and the action potential for any human being is constant throughout the life. Like the shape of your action potential is going to be same when you're a kid and when you'll be adult.
I see. So now we are using a square pulse instead of that action potential.
No. The thing is about stimulus. There are two types of stimulus. There is a diphasic and mono. So you can provide a single pulse or a single positive and a negative pulse, so that less power is required. And the shape and size, that is the pulse width and the amplitude, they decide the amount of action potentials that will be generated in the nerves.
See, I told you I learned a lot from my thoughts.
But will the muscles react differently if we have a different shape
Yes,
of the signal?
see, the muscles react to the rate of action potentials and not the shape, because as I said, the shape of action potential remains the same entire life. The information is carried in the bit rate, the rate at which the action potentials reach your muscle fibers. So yeah, that depends on it.
Thanks.
This happens always. So.
Quick question. One thing is about the coating that was probably used for the micro electrode. The coating.
Utah electrode coating. Yeah.
Okay. Time to dance.
What was the coating used on the electrode to make it biocompatible? Second is, what benefit did we have to make it slant?
Okay. I don't remember the details of the coating. I know that there is a coating, and I know people have tried different things, and they're happy with what they have now, but I don't know the details. But these are all published work, so you can actually look it up. Okay. I don't. Again, the different lengths. Remember when we talked about fascicles that are close to the implant site and away from the implant site. So you want to access the fascicles, the bundles, both away and close. Okay. The shorter ones will actually access the ones near the implant, and the longer one will access the ones farther away from the implant.
Okay, so it is
It's
not
the
like
tips that matter.
It is not perpendicular to
It
the
is
fascicle,
actually
but
inserted
slant.
is.
perpendicularly.
It is.
It is inserted that way.
Okay.
What is used as the ground reference for the probes?
Oh, okay.
Because you gotta have, the current's gotta go somewhere, right?
Yeah. I was asked this question sometime before, and I went to my daughter, and she actually told me the answer. I believe that, yeah, go ahead.
Maybe I can answer.
Yeah.
So the ground is actually the saline, what we can say, the fluids that are outside the nerves, that is the nerve fluid, that provides the reference ground for all this action potential measurement as well as the stimulus that we provide.
So is an electrical connection made to that?
Yeah, so there are two electrodes. There is one for the stimulus and the other one measures the ground, which is actually outside. It's called the extracellular electrode, which tracks the ground potential, and that's the reference ground for all the measurements.
And how about the impedance? How much current do you have to drive into the probe? I mean, that sort of
Oh yeah,
stuff.
that actually depends on the salinity of the fluid as well. So that is specific modeling depending on what the individual subject is. Differs from person to person.
I'm glad these people are here. These are kind of questions that I don't even care about. I just need signals, so yeah. But
That's why we have students.
right. No. This is an area that is a little different from what I grew up in, right? In the sense, so I'm learning with all these things, and so it's a good thing.
You can call them an analog
Do the-
person.
Yeah. Right.
I could design a digital filter if my life depended on it right now.
Oh, well, with analog filters, I probably can say similar things too. So,
Maybe
anything else?
would come from the digital world.
They're looking
Need
for
to-
engineers.
Anybody who's looking for ground, there's an
do.
analog.
Get a job, it's late to send out engineers.
By the way, about the ground, I knew that there are a couple of electrodes that is used as ground, but you get much more details than I knew. So that's good.
Okay, any other questions? If no, let's thank our speaker here.